In today's lecture, we learn about the external covering of the cell, that is cell wall and the cell membrane. Cell wall is the outermost covering of cells, but not all types of cells have cell wall. For example, plant cell, bacteria and fungi do have a cell wall, but animal cell does not have a cell wall. Now, each type of organism has different composition of cell wall depending upon their function and the environment they have to face. For example, plant cell wall is made of cellulose, bacterial cell wall is made of peptidoglycan, and fungal cell wall is made of chitin. The cellulose is a carbohydrate, peptidoglycan is a combination of protein and carbohydrate. The word peptido came from peptide, that is a long protein chain, and glycan is for the glucose carbohydrate. Chitin is nitrogen containing carbohydrate. The cell wall functions to protect, to shape, and to hold the plant against the gravity. It provides protection because cellulose is an indigestible substance that cannot be absorbed or digested by all sorts of organisms. So not all organisms eat every plant. It provides shape by not allowing water to seep inside the cell so the cell does not burst and change the shape much more frequently. It is a rigid structure, so it holds the plant against the gravity. Now, there are two types of cell walls, the primary cell wall and the secondary cell wall. Between these cell walls, we have the middle lamella. It is made up of calcium and magnesium salts and pectin. Pectin is a polymer of amylose, which is also a carbohydrate. Now let's talk about different features of primary and the secondary cell wall. The primary cell wall is located next to the cell membrane, while the secondary cell wall is the outermost layer. So coming from inside to outside, there is primary cell wall, then the middle lamella, and then the secondary cell wall. The primary cell wall is thin and flexible, that is, it allows the cell to move, whereas the secondary cell wall is thick and rigid. It provides the integrity, it maintains its structure of the plant cell. The primary cell wall develops during the cell division, that is, it is important for the growth of plant, whereas the secondary cell wall develops when the cell reaches its maximum size. So after the development of secondary cell wall, cell cannot grow further. Next is the composition. The primary cell wall is composed of cellulose microfibrils, hemicellulose, and pectin, whereas the secondary cell wall is composed of cellulose, hemicellulose, lignin, inorganic salts, and pectin. Lignin is the molecule that provides the rigidity to the secondary cell wall and maintains the structure of the plant cell. Function. Primary cell wall provides strength and plasticity or the elasticity to the plant cell, whereas the secondary cell wall provides shape and mechanical support. Next, we'll discuss the cell membrane. Cell membrane is present in all organisms. It is present inner to the cell wall in the organisms that contain cell wall, but it is the outermost covering of an animal cell. It controls the passage of materials in and out of the cells to maintain a pH and ionic concentration for proper functioning of enzymes. Cell membrane is composed of proteins 60 to 80%, lipids 20 to 40%, and carbohydrates in small quantities. The arrangement of these elements is explained by the fluid mosaic model. This model states that the cell membrane has a jelly-like consistency, hence called fluid, and the proteins are present as beads or the icebergs floating on the water, hence called the mosaic. The major component of cell membrane is made up of phospholipid bilayer, that is, there are two layers of phospholipids that are almost 7 nanometer thick. Now, what is a phospholipid? Phospholipids are the lipids that is made up of glycerol and fatty acid with phosphate groups attached to them, hence called phospholipids. 3 glycerols and 1 fatty acid combine to form one molecule of lipid. Now, the phospholipid molecule has two components, the hydrophilic portion and the hydrophobic portion. The term hydro means water and philic means loving. So, the hydrophilic part is the part that loves water or it can adhere to the water. Similarly, the word hydro means water and the phobic means hating. So, the hydrophobic parts that hate water or they cannot survive in the watery conditions. As we know that human body is 60 to 80% water and all the cells and their cell membranes are present in an environment containing large amount of water. So the hydrophilic portion faces outside and the hydrophobic portion are present inside. They face each other. The hydrophilic portion is also called heads and the hydrophobic portion is called the tails. Each phospholipid molecule has one head and two tails. Between the two phospholipid layer is present the cholesterol molecule, and between the two phospholipid molecules is present the protein molecule. The protein molecules are actually glycoprotein, which means that there is the protein molecule that has attached a carbohydrate chain to it, hence called glycoproteins. These glycoproteins perform a number of functions within the cell membrane that we'll look into later. On the external side of cell membrane, the phospholipid layer heads also have a moiety attached to them called the glycolipid layer. Filaments of cytoskeleton are also present on both sides of the plasma membrane. Now, if we divide the plasma membrane into two halves, that is the left and the right half and the up and the lower half, then we can see that the right and the left half, they are not identical, and the upper and lower half, they are also not same. For example, the right half is made up of phospholipid molecules and the glycolipid chain attached to them. However, the left half is made up of the glycoprotein and the phospholipid molecules. On the upper half, we have the glycolipids, the glycoproteins, but there are no glycolipids or glycoprotein present in the lower half. Also, there is one cholesterol molecule in the upper half. However, there are 1, 2, and 3 molecules of cholesterol in the lower half. Hence, the plasma membrane is asymmetrical. The two halves are not same to each other. Coming towards the function, the lipid part controls the fluidity of the membrane, mainly the unsaturated fatty acids. Cholesterol provides strength and controls the movement of polar, that is the charged molecules, through the plasma membrane. Proteins perform a number of functions. They can be channel or carrier proteins. They act as enzymes, receptors, antigens. The glycoprotein and glycolipid act as cell surface marker. Channel protein is a protein that allows the passage of molecule through itself without changing the configuration or the shape of itself. However, a carrier protein is a molecule that attaches to the molecule and change its shape. First, the hilum is present in the center, the curved portion, and later on the curved portion is present in the lower half. In this way, it allows the molecule to pass through it. It occurs in a similar fashion that if we stuck a piece of candy inside a straw and then apply pressure from both sides using our thumb to move the piece forward. In short, a channel protein passes the molecule by simply forming a channel, whereas the carrier protein attaches the molecule itself and carries the molecule towards the other end of the membrane. Now, receptors are the substances to which certain molecules attach. We'll discuss in detail about the receptors and the attachment in the next topic of the signal transduction. Antigens are the molecules that act as surface markers or the signboards of the shop. Just as the signboard of a shop tells about the name of shop, the antigens will tell about the type of the cell. Glycoproteins and glycolipid act as cell surface markers. That is, they are present upon the surface of cells and help them to identify the similar cells and attach to them to form the tissues. Earlier, we said that the function of cell membrane is control the passage of materials in and out of the cell. Passage, or in other words, we can call the transport. There are different types of transport that occur through the cell membrane, namely diffusion, facilitated diffusion, osmosis, and active transport. One by one, we'll look into the detailed mechanism of these transport systems. Diffusion. Diffusion is the passage of molecules through the semi-permeable membrane, that is a membrane containing pores, from an area of higher concentration to the area of lower concentration. This type of transport occurs for the non-polar molecules like oxygen, carbon dioxide, and water. Next is facilitated diffusion. As the name suggests, this type of diffusion is facilitated or supported by some other molecules. These are the channel proteins or the carrier proteins. Channel proteins simply provide a channel for the passage of molecules, whereas the carrier proteins themselves carry the molecule to the other end. So, the facilitating molecules are the channel proteins and the carrier proteins. As this is also diffusion, so it must occur through a semi-permeable membrane. This semi-permeable membrane is the plasma membrane. Hence, we can conclude that plasma membrane is semi-permeable. That is, it permeates or allows some molecules and does not allow other molecules that are unsuitable for cell to pass through it. Now, this type of diffusion occurs for the water-soluble substances because the inside is hydrophobic. These molecules cannot easily cross. So, the proteins help to pass them through the plasma membrane. These molecules are sodium, glucose, and the polar or the charged molecules. Now, channel proteins can be voltage gated or ligand gated. The term gated means guarded or protected. Voltage gated means that these proteins are controlled by the ion concentrations on both sides of the membrane. And the term ligand gated means that these can also be controlled by the concentration of ions present on both sides of the membrane. The carrier proteins are controlled by the temperature and the saturation. Saturation means the concentration on both sides of the membrane. How many carrier proteins have been occupied by the membrane? This is called the saturation. If all of them are occupied, then the saturation is maximum, and if only one or two is occupied, then the saturation is minimum. They are affected by temperature because temperature changes the structure of proteins, and the proteins have specific charges inside them that can bind to the coming molecule and transport them. So the temperature changes the structure of protein, and hence they cannot bind to the coming molecule and cannot transport them. So they are affected by temperature and saturation both. Osmosis. Osmosis is similar to diffusion, that is, molecules move from area of higher concentration to lower concentration through a semi-permeable membrane. But the difference is that these molecules are dissolved in some sort of solvent, and inside the body this solvent is mostly water. The solute molecules can cross, but the solvent will not. Now, as the passage of molecules starts, a time will be reached when the concentration of molecules on both sides will be equalized. That is, equal number of molecules will be present on both sides, and at this time the pressure on both sides is called the osmotic pressure. Osmotic pressure is the pressure at which solute concentration on both sides is equalized. An easy example of osmosis is that when seeds are soaked in water, they swell after some time. That is, the water from outside is the solutes dissolved in the water present in the surrounding starts to enter the seed and swell the seed eventually. Active transport. The previous type of transports, that is diffusion, facilitated diffusion and osmosis, occurs from the higher concentration to the lower concentration. But active transport occurs from lower concentration to the higher concentration. Hence, it will require some energy to move molecule against the concentration gradient. The concentration gradient is the difference of molecules on both sides, and along the concentration gradient means from high to low, and against the concentration gradient means from low to high. So, if the molecules are to move from lower concentration to the higher concentration, they would need some energy. Now, there are two mechanisms to provide that energy. One is that ATP molecule breaks, it releases one phosphate, and the energy is released by the bond breaking. This energy is used to transfer the molecules. This is called primary active transport. An example is sodium-potassium ATPase pump. That is, by using the energy of ATP, a pump or the energy is created, and it will transport both sodium and potassium against the concentration gradient. Another type of active transport is secondary active transport. In this type of active transport, one molecule moves from lower concentration to the higher concentration, and the other molecules moves from higher concentration to the lower concentration. Hence, the energy released by the movement of molecules from higher to lower concentration is utilized by the molecules moving from lower to higher concentration. The example of this type of transport is sodium hydrogen antiporter. Porter, that is, it is a transporter. Antiporter means that it is transporting the molecules in opposite direction. One molecule is coming inside the cell and the other molecule is going outside the cell. Sodium is moving inside the cell and hydrogen is moving outside the cell. One molecule is moving along the concentration gradient from higher concentration to lower concentration, and the other molecule is moving against the concentration gradient, that is from lower to higher concentration. So energy released by hydrogen transport is utilized the energy required for the sodium transport. To summarize, cell wall is the outermost covering in plant cell, bacteria and fungi, but it is not present in animal cell. Plant cell wall is composed of cellulose, bacterial cell wall is composed of peptidoglycan, and fungal cell wall is composed of chitin. Cell wall protects the cell, provides the shape and hold it against the gravity. There are two types of cell wall, primary and the secondary cell wall, between which is the middle lamella. It binds both the cell wall together. The primary cell wall is composed of cellulose, hemicellulose and pectin, and the secondary cell wall is composed of cellulose, hemicellulose, lignin, inorganic salts and pectin. The primary cell wall provides strength and plasticity to the cell, and the secondary cell wall maintains the shape and provides mechanical support. Cell membrane. Cell membrane is present in all organisms. The cell wall containing organisms, it is present inner to cell wall, and it is the outermost layer of an animal cell. It controls the passage of molecules by multiple mechanisms such as diffusion, facilitated diffusion, osmosis, and active transport. Cell wall structure is explained by the fluid mosaic model. That is, it is made up of phospholipid layer 7 nanometer thick. It has the glycolipids, glycoproteins, and cholesterols attached to it. Inside and outside cell membrane are present the filaments of cytoskeleton. The cell membrane provides fluidity, strength, movement of polar molecules, The proteins perform a number of functions. They can be channel or carrier proteins, enzymes, receptors, antigens. Glycolipids and glycoproteins act as cell surface markers. Diffusion occurs through a semi-permeable membrane along the concentration gradient, and it occurs for oxygen, carbon dioxide, water, and the non-polar molecules. For the water-soluble or the polar molecules, we have the facilitated diffusion, a type of diffusion facilitated or supported by the channel and carrier proteins. Osmosis occurs for the solvent and the solute mixtures, where the solute molecules move through a semi-permeable membrane along the concentration gradient. The pressure at which the concentration on both sides is equalized is called osmotic pressure. Active transport is the movement of molecules against the concentration gradient. It can be primary or secondary active transport. Primary active transport uses energy from the ATP molecule, and the secondary active transport uses the energy of the nearby molecules.